Morning, there is a question. Did you have something, Stephanie? Maybe. Okay, your microphone's just on. Okay. All right, so let me break you up into the lymphatic questions, and I am going to have one group that's the first couple questions in the immune system. So we may not get to you guys, and don't worry, on not Tuesday in lab, we'll get to you because the lecture will be held during the lab. But we might get to you, so we'll see. So let me find the breakout rooms. Let me make 5, manually put people in, create the rooms. I'm going to give you, let me look at these questions. Probably 15 minutes is fine in the room. And I'm going to rename the first room will be, I'll just call it lymph questions 1 and 2. So you're doing those first 2 lymphatic system lecture questions. And then I'll rename the second one lymphatic. You're going to be doing 3 and 4. How I'm breaking these up is these are related questions to each other. So that's why you'll see some people have more questions than other people. As you'll see, this group has lymphatic 5 through 8. And yes, and then the next one we're going to rename as lymphatic 9 through 11. Yes, and this last group we're going to rename as immune. I think I just said 1 and 2. Okay. All right, so I did post these questions in the announcements. So if you have, since they're in 2 different documents, if you're confused, you can always open the announcements on Canvas and you'll see the questions. All right, so let me assign some folks, and I'm going to do it pretty randomly, except I think group 1, or the immune 1 and 2, I'm going to make that a group that is all together, so I know who you are when you come back. I'll probably just pick the first 6 people, 2, 4, 6. Okay, so I'll pick the first 6 people on my list for that last group, so I know who you are when you come back. Okay, let me do the lymphatic. I think I did that right. Wait a second. No, I didn't. Oh, no, I didn't do that right. Dang, sorry. I gotta go to this one and assign, and then I'll go to the first 6 people. Okay. All right, now we'll go to the other ones, and I'm just so you meet each other, you know each, don't always work with the same folks. I'm going to kind of mix you up. So maybe I'll go every 3rd person. All right. And then I'll go to the next group. Okay, and the next group, 2, 4, 6, 8, 10, 12. I probably have to add a few people to some of the other groups. And one more, where is it on here? Yeah, assign 2, 4, 6. And maybe we'll just, since there's 3 questions in this one, we'll just put more people in that. Okay. All right, so Yeah, 6. There's one group with 5. Okay, that looks good. All right, so I'm going to go ahead and open the rooms, and if you have any questions, just come back. And remember, you need to assign a spokesperson to talk about your lecture questions when you come back. All right, we'll see you in about 15 minutes, unless you... So, because I know we did start with one more person, but, oh, there you go. There's somebody who just came in. Okay, so the first group on the lymphatic, which are doing questions 1 and 2. So this is not the immune first group, 1 and 2, but the lymphatic, where they're going to be talking about the types of circulating white blood cells. Do you guys have a spokesperson?
Yes, Professor. I'll be taking number 1, and Ileana will be taking number 2.
Okay, great. Thank you, Kelsang.
Thank you.
And let me share my screen with you, because I'm going to just kind of take notes on this part. And this is really going to be something we're doing in lab as well. I don't know why it does that sometimes, but okay, here we go. All right, so we have the types of white blood cells already out here for you, Kelsang. So can you tell us about the 5 circulating types of white blood cells? So what they're talking about when they say circulating is that they are actually in the bloodstream.
Yes. So neutrophil is the cytotoxic. It kills self and non-self cells to protect from pathogen and act as immune.
Okay, so that is one of its functions, but it kills, when it's killing, it kind of has 2 ways to kill. So what 2 ways can it kill things? Let me give you a hint. One of them is it consumes it. Does that help, Kelsang, or anybody else in that group?
A phagocyte, the numerous circulating phagocyte.
Yes, it is phagocytic.
Oh, phagocytic.
And it also can kill because it releases stuff that's toxic. What does it release?
It releases, it releases enzymes, cytotoxic.
Yes, good. Enzymes and other antimicrobials is what we'll call it. Enzymes, and we'll talk about more specifically who those are.
Okay.
Let's see if I can fit the word antimicrobial in there, because I didn't give myself a lot of room. Now, I put these in a certain order because, as your lab manual tells you, there's a trick to knowing the most common of these white blood cells to the least common. These percentages you're seeing, we're going to use in lab today. These are the normal percentages of white blood cells in a differential white blood cell count. So the most common are neutrophils that make up more than half of all those circulating white blood cells. Okay, so the next one, by the way they help you remember this is never let monkeys eat bananas. So that's a memory trick to try and remember unfortunately, the order. Sorry. The percentages you'll be learning for lab, not for the lecture. So, Kelsang, what about the lymphocytes? Maybe I'll change color.
Lymphocyte, it is an adaptive immune system, and it has a B and T cells, and it also has an NK cells, which are of innate response. And NK cells are a circulating cells, and then the blood. So in the blood, if there is a circulating lymphocyte, it is the immature B, T, and NK cells together, not only the NK cells as a separate. And then B and T cells get matured in the lymphatic system. and NK cells maybe somewhere in the connective tissue. And
Okay. All right. So circulating, if there are lymphocytes, which you see, there are quite a lot of lymphocytes. Can make up almost half of the remaining circulating white blood cells. As Kal Sang said, these are often on their way to somewhere. And if they're an NK cell, they tend to go out of the blood. I'm sorry, I got a blue jay. They tend to go out of the blood, and so the numbers that are actually circulating the blood that are NK cells is probably fairly low, because it's just the replenishing of the supplies in the connective tissues, just like Kal Sang said. That's also true for the B cells. Of the adaptive response, there are two subtypes of lymphocytes, B and T. And generally speaking, B cells are in the blood only as they leave the bone marrow and then are on their way to lymphatic tissues, just like Kal Sang said. So the majority of cells that are circulating in the blood are of the T type. Because as you'll see, they not only end up in lymphatic tissues, they also patrol the body. What did you just say?
The mature T cells are in the thymus.
Yes, and they do have to go to the thymus in order to become mature T cells. You're right. So some of them that leave the bone marrow are heading to the thymus to mature. Okay, so what about these monocytes?
Okay. Monocytes, it releases cytokines, and then it is circulating around immature macrophage and dendritic cells.
Okay, so one of their big jobs is they release cytokines, and we haven't defined that, but cytokines are part of your immune response where you're signaling. So you could be signaling locally, like a paracrine response. You could also be signaling at a distance, but these are messengers, as it were, chemical messengers from one part of your immune response to another part of your immune response. That doesn't mean just between white blood cells. So inflammation, that we'll start talking about next week, is actually affecting the smooth muscle and the endothelial cells of the blood vessels to allow inflammation. And that's triggered by cytokines. So that is a big role of these guys when they mature, because the one cell that is definitely immature, well, it's circulating. You can't tell with a lymphocyte whether you're dealing with a mature or immature cell, but if it's a monocyte, it is immature. And just like she said, it can become either a macrophage, which I probably can't fit in one line, or it may be a dendritic cell. And we'll talk about those different cell types. One of its other roles besides cytokines is it also kills. And how does that kill?
Okay. Monocytes.
Let's see if you've got
it. Kills.
The hint is in the name of you can't see. I can't see where it is on here either. This name.
It's a monocyte become mature to macrophage. I'm not sure on this.
Okay, what is the macrophage name suggest it does?
Immature. Oh, what it does?
It's phagocytic, right? Phage, that's telling you phagocytic. And in fact, the word macrophage means big eater. So as we'll see, these guys are the most effective of the phagocytic white blood cells. They also last a long time. They're very large cells, so they can eat larger things than, say, our neutrophil here. So Dendritic cells, I know it's not in the name, but they are also a phagocyte. So all of the monocytes are going to become long-lasting phagocytes, which, if they're a macrophage, can really consume some of the larger things that they might have to consume. Okay, going on. So let me see if I come up with another color. Eosinophils, we're not going to hear a whole lot about. So if you didn't find a lot, Cal saying that's fine. Do you have something on what eosinophils do?
Yes, a little bit. It releases hydrolytic and pro-inflammatory against parasitic worms and during the allergic responses.
Yeah, so these, you're starting to see something you're going to deal with today. These are highly specialized, and what we see is that their numbers rise. They're no longer just 2 to 4%. If you have a parasitic worm infection or an allergic response, you tend to see a rise in the number of these eosinophils, the percentage of these in that person's bloodstream. So our book doesn't go a whole lot more beyond what Kelsang just said about eosinophils. So that's all you'll really need to know. I do think in micro, once you take it, they do talk a little bit more in micro about eosinophils, but so you'll have to hold off for that. I'm just going with how our book does it. All right, our last cell here. And let me pick a different color, maybe pink, are basophils. So
Okay.
what do they do?
But sure. Basophils releases histamines and heparin in chronic inflammation and allergic response. And heparin acts as what you call that one? Flush the pathogen contaminated blood during the active bleeding.
So they do it, that does it by being an anticoagulant. So it blocks the actual blood clotting, not completely, but it slows it down. And that allows, as she said, you to bleed for a little longer with a cut. So then you might, if you had no heparin there and no basophils releasing that heparin, so that you could flush away. So using your bleeding as an outward flow to flush away pathogens that might otherwise get in the bloodstream. So these, you're going to be drawing today, these 5 white blood cells, and recognizing them in smears of normal blood. So you'll end up with, well, it'll depend. Some of the pictures have more than one, or one of the pictures has more than one white blood cell. You'll either have 4 or 5 drawings of these just normally, and you should be able to recognize them. And we'll talk in lab how you can tell them apart, but there are some distinctions, especially in how their nucleus appears. And also, as we'll see in the next person's, them being either a granulocyte or an agranulocyte. Okay, so who was doing question 2? I've forgotten already.
Ileana.
Okay. All right. So Ileana, have you got something on the lineage of the white blood cells and granulocytes versus agranulocytes?
Yes. They come from I believe both of them come from the myeloid lineage.
Yeah. So I put this here to help. So you could just. This is
awesome.
Okay.
Yeah. So the monocytes are agranulocytes, and I think the rest of them are all granulocytes. And
the rest of them in that lineage, you mean?
Yes.
So that's right. Oops, sorry. These guys are agranulocytes. And these guys are all granular.
Granulocytes. Yes.
Now, who else is a granulocyte that's not in the myeloid lineage? So there's only one other lineage besides
Yeah,
myeloid.
the lymphocytes.
Lymphocytes. These are also agranulocytes.
Okay.
Okay. So I reproduced this from, in some ways a nicer picture, but in some ways a confusing picture because it has so many more rows on page 655. So remember the hemocytoblast is the mother of all the stem cells. This is sometimes called the hemopoietic stem cell. And it's residing in the bone marrow and generating a myeloid stem cell. This is still a stem, so it can divide and then it can replenish itself constantly. And you might recall that it makes proerythroblasts that make red blood cells, megakaryoplasts that make platelets. And what decides it to do that is if you have EPO, which you always have some because you're always making red blood cells, but they are listening to EPO. And here it's listening to thrombopoietin. So what about for the other lineages? What regulates the rate at which they make these other lineages? There's sort of an umbrella term. Do you know what it is, Ileana?
Is it the colony stimulating factors?
Right. So colony stimulating factors, which again have that abbreviation that can be confusing.
CSF.
Yeah, but I don't have room to write out colony stimulating. So are going to include EPO, thrombopoietin, because the colony they're talking about is the choice of the stem cells in the bone marrow. So the hemocytoblast is making these, and under the influence of these guys may make more myeloid. The others we're going to call just colony stimulating factors, but there are many of them. But the ones we're going to fall and talk about in our immune response are called interleukins, which we abbreviate ILs. So good. And it says that all the granulocytes, what Liliana said, all these granulocytes are all from a stem cell called a myelioblast versus a monoblast or a lymphoid stem cells, which are agranulocytes. So
Yes.
What else did it ask here? That's about it. That's the answer. For those.
And I just wanted to add, I have a note on my questions here. It says the signaling molecules trigger the formations of each type of cell. And since they all come from the same stem cell, if it makes more of one type, it makes less of the other type.
Perfect.
So, yeah.
Yes. I'm so glad you said that because that's what we're doing in lab again. So remember, let me roll up here. If your colony stimulating factors say you've got, a parasitic worm infection and this percentage goes up, well, this has to add all up to 100 in any differential white blood cell count. So if this becomes high, then there's got to be a decrease in the others. And that doesn't mean that in your sampling of your blood, generally, especially something like an eosinophil, you're going to be weakened by having fewer of these. But yes, as she brought up, this cell has to make a decision. It's balancing all these colony stimulating factors so that there's only limited resources. It can only do mitosis at a certain rate and make a decision as to which way the one daughter cell is going to differentiate. So if you're making many more of the granulocytes, say, you're really stimulating these myelioblast lineages, then you may see fewer of these, or even fewer of these. So the other thing you'll be doing in lab today is drawing a leukemia slide. The particular leukemia is lymphocytic, so it's a leukemia along this lineage. And what happens in those patients is they're generating so many of these that you see fewer of these, and even eventually, as the person gets sicker, fewer red blood cells, and their blood becomes very pale. Because there's so few red blood cells. Because clearly, this is making 2 million a second, so you're always making red blood cells. It's going to be diminished if you don't have enough materials. Okay, great. So let's go on to the second group, and I didn't make any figures for that group, so I'm going to stop sharing. That you can find those figures, and you can find the numbers you'll see in the lab. As far as the functions of the white blood cells, we're going to go into quite a bit more detail about them and who are the cytokines and how they go about doing their jobs as we go through the immune response in more detail. So who is the spokesperson for the second group?
That will be me, Jeanette.
Okay, Jeanette. All right.
Okay, so the question was, describe the adaptations of the lymphatic system in its role of monitoring blood, lymph, and interstitial fluid for pathogens. And we started discussing the stroma that is formed by reticular connective tissue. And the stroma will act by serving as a net that captures pathogens in the blood to be targeted by lymphocytes and macrophages. We do have lymphatic organs are surrounded by a fibrous capsule, while lymphoid tissues are actually embedded in organs under the epithelial layer, which will be a part of the next portion, which is MALT. MALT stands for mucosa-associated lymphatic tissue, and this is found along mucous membranes where most infections begin. So your examples are the digestive, respiratory, and urogenital system. And if you want to see an example of MALT, there are Peyer's patches within the intestines, and these patches are basically just aggregated lymphatic nodules of MALT.
Very good. Okay, so I just wrote a few notes, and I'll share those. So there's a few words used which you may not be as familiar with. Let me share the screen again. Just to make sure everybody kept up. Okay, she used the word stroma. So stroma occurs in connective tissues, and remember connective tissues, what is going on with that? Connective tissues have fibers, and I'm sure you remember collagen because we talked so much about it in 103A. And elastic fibers, which we also talked a bit about, but one that we didn't talk a whole lot about, but we're going to encounter today are reticular fibers. These are fibers in the extracellular matrix of connective tissues, well, of any extracellular matrix. So extracellular matrix fibers. Any connective tissue can have any mixture of these. And if it's a fibrous connective tissue or a irregular, dense, irregular, or reticular connective tissue, you might remember all those different connective tissues, or areolar. They're certainly going to have reticular fibers, especially the areolar reticular connective tissue, dense, irregular connective tissue. Perhaps dense, regular, or bone, that's not going to have much reticular fibers. Why you want these is these are sites that cells can reside on. I'll just put hold cells. And the cells we're talking about are going to be these white blood cells that have left the blood and are waiting around in the stroma, just as Jeanette said. And these fibers also act kind of as a filtering device, like she said. So not only are you physically filtering the interstitial fluid, the lymph, or the blood, as we'll soon see, I think, in the next question, You have the fibers doing a physical filtering, and then sitting on those very fibers are going to be immune cells. So that's good. MALT I put down as just a definition, and that's going to be part of your lymphatic system, but these things are called lymphoid. So if you see the word lymphoid, oid means false. That's what that suffix means. It's not really false lymphatic tissue. It's not an organ. So if something is an organ, that means you can go in and you can dissect it out of the body because there's fascia that's separating it from the other organs around it. If it's lymphoid, you can't physically dissect it out, because if you were, you'd be cutting up that particular organ. So basically embedded in these three lymphatic systems, three mucosal systems, you have lymphoid tissues. So what makes these guys mucosal system, we're going to talk about in lecture a little bit more, probably next week when we do our first mucosal membrane, which will be our respiratory system. Next week, maybe a little later than that. But I'll briefly talk about it today in lab. Okay, great. So what about these? Let me take notes as is this still you, Jeanette, doing the next question?
I think number 4 was exactly worded the same as number 3. They were the same questions.
Our group was doing numbers 4 to
Yeah. So
Did I mess up and give two of you group 4
or 4.
no, in the lecture questions, number 3 and 4 is the same exact question. I think it's just an error.
Really? Because I have my printed copy and it says, what are the functions of the lymphatic organs and structures? Is that not what you
Oh, that's number 5.
Oh, so somehow things got renumbered.
Oh, it's just
No.
that question 3 was just redone to question 4. It's the same exact question.
Yeah, I think it's Word, how it automatically numbers stuff. So I think I just didn't notice it had renumbered. So is anybody dealing with the functions of the lymphatic organs and structures at all?
I'll be talking about that.
Okay, Jessica, why don't we'll just label this functions, lymphatic organs.
So I'll be talking about numbers 4 to 6, and then
Okay.
Sana will be doing 7 to 8 for our group. So number 4 is, what are the functions of the following lymphatic organs? So for the nodes allow lymph to pool in them before exiting, which gives a chance for B and T cells to detect the pathogens. The spleen monitors blood. So there's red pulp and white pulp. The red pulp recycles damaged red blood cells, while the white pulp includes B cells, T cells, macrophages, and dendritic cells that screen for pathogens and make antibodies. And then the thymus is the site of T cell maturation and selection. The bone marrow is where B cells are generated. And then the MALT are secondary lymphoid tissues that are found in the respiratory, urogenital, and digestive systems, and they initiate immune responses in those certain areas.
Okay. Good. That was perfect. I'm going to just put up here, filter blood. So you have a bunch of body fluid circulating in your body through your bloodstream, but you also have lymphatic fluid. So shortly, we're going to be talking about how the lymphatic capillaries can pick up larger items than your normal continuous capillary, and therefore they could pick up infectious organisms. So you have nodes periodically along these lymphatic systems, vessels, that will then allow, as she said, lymph to pool in them. They're made of reticular connective tissue. In fact, all of these guys are going to be made of reticular connective tissue. And then, of course, the bone marrow is made of the stem cells. So they're probably sitting in reticular connective tissue, but I've never seen it said that. So anyway, that means that they have cells, as she mentioned, B and T cells and dendritic cells for that matter, that are sitting there and they're surveying the lymph. That's what they're looking at. The spleen, because you have another circulating body fluid, blood, is going to be similarly filtered. Now, most of the spleen is looking for dead and dying red blood cells to recycle them. But islands of white pulp, it's kind of a strange name. These are all reticular connective tissue, whether they're red or they're white pulp. But the white pulp has the B, T, and dendritic cells. That's what I mean by ditto up here. They are surveying the blood. So this guy surveys the blood for pathogens or antigens, as we'll soon see. Bloods. Okay, the thymus we'll come back to in a second. We have one more body fluid that's useful to filter if you're right at the area of most likely getting an infection. So as we'll soon learn, our skin is a really good barrier. Most infections, and you probably know this from just having lived it, come in through your mucosal membranes, your respiratory system, your digestive system, and hopefully less commonly, your urogenital system. But probably most of the things you've gotten sick with have either been a digestive tract or a respiratory tract. So you have embedded all through these mucosal membranes some lymphoid tissue, and that's part of MALT, and it's actually filtering the interstitial fluid right at the border of the epithelia. So it's right under the epithelia, and it's therefore if a pathogen comes in, gets through that protective epithelial lining, you have some B and T cells. So these are, again, they're going to be B, T, and dendritic cells that are then surveying that interstitial fluid. Now, I skipped over the other two because they have more roles in the generation of bone marrow of all the white blood cells and the red blood cells for that matter, and platelets. And then the thymus, which helps select, and I think I wasn't fast enough, and mature the T cells. And we'll talk about that process when we get to the immune system. Okay, so who is doing 5?
I'm going to do 5.
Okay.
So number 5 is name 2 sites where crypts are located and explain their functions. So the 2 sites are the tonsils and the appendix.
Okay. So what is a crypt?
They are enfoldings on the surface of mucosal membranes that allow B and T cells to culture the microbes that enter in order to educate the adaptive immune system.
Right. So if you looked at your tonsils, which you know are kind of enlarged areas, if you look in the back of your throat, then you'd find along that surface epithelia, it would be highly enfolded, like she said. So you'd have saliva out here and, food or fluid that you're drinking. And these infoldings, whenever you see that, what are you trying to increase?
Surface area.
Surface area. You're also allowing little microbes, I'll make them a different color, to actually grow within these little infoldings. And then right underneath that infolding, you have malt. You have some lymphoid tissues. And there's B and T cells and dendritic cells in here. And they're going to be surveying this, what you're cultivating, so that you're always mounting a adaptive immune response to organisms that are coming in or are being chronically present, because as you probably know, you have microbes all along your oral cavity, and the appendix is right next to the large intestine, which has probably the richest population of microorganisms that we cultivate within our body. So this is not only mounting it to a pathogen that might just happen you be exposed to, but it's also mounting some level to the normal residents, because you want to make sure you keep them at a respectable level. Okay, so this, I was just looking at the chat because the one new one showed up. All right, those are crypts. What body systems are lined by mucosal membranes? I think we already answered 6, right? That was the respiratory, digestive, urinary, and reproductive systems that are mucosal membranes. So moving on to how these lymphatic capillaries work, and I don't have, maybe the numbering is off for me. Who is going to talk about the one that says, describe how the endothelia form?
That would be me.
Okay.
So this question's asking us, how does the endothelia differ in the lymphatic capillaries versus the circulatory capillaries? And that is that the endothelia in the lymphatic capillaries are held together tightly, but they're actually joined loosely, which gives them their flaps, and that allows lymph to flow in a one-way direction. So it allows fluid to enter but not exit. And if pressure inside the capillary is greater than the pressure outside, then the valves are forced shut. And if the pressure inside the capillary is less than the pressure outside, then the valves open. And then the capillaries also have collagen filaments that attach them to the surrounding structures, so that if the fluid builds up outside the capillaries, then the flaps can open, as opposed to collapsing if the fluid was building up outside and they didn't have the collagen filaments. And then the thing that also makes them different is that they're able to take in larger particles and proteins for filtering, as opposed to the blood capillaries, which cannot.
Okay, that's nicely done. So this is a blood capillary. Remember, there is some paracellular movement, right? This is paracellular. So that you have, and I know we just had the test, so you should remember that generally speaking, you have about 10 going outward, right? And you have about 8 going inward, which means left out here, there's 2 millimeters of mercury, even under just resting conditions. And what I mean by resting is, on average, it's going to be always leaving some interstitial fluid, and that's going to accumulate out here as interstitial fluid. And as it does, you're going to start to see swelling to occur. So when swelling is occurring, let me make that different. As this swells, because it keeps accumulating this, these collagen fibers start to be pulled apart. Since there's an anchoring protein, as Sana said, so this is, I'm going to call it an anchor to the collagen protein. They don't name it. That is a membrane protein on the endothelial cells that make up the lymphatic capillary, which is in blue. That means that the outer flap one will start to move outward with swelling. Oops, that's the wrong direction. So these will swell and move outward, and they're going to open this area wider. So they have no tight junctions between their adjacent endothelial cells. Whether or not fluid moves easily depends on how much interstitial fluid is around them. So as you do your normal capillary exchange and you accumulate that 2 millimeters of mercury, you get some slight swelling that opens this, and maybe I'll make lymph a different color. So, well, that's the same as that. So that lymph will flow into here. And as it does, what happens is this value keeps decreasing, right? You keep getting less and less of it. As it decreases, these guys move back and close these mini valves. So, so that's how lymph enters. And as Sana, oops, that's nice. As Sana said, that means why lymph cannot leave once it enters. It can't go out, can't go in the other direction. It's a one-way direction. And they call these overlaps mini valves. I think it's really super clever, if you ask me. You know, that you anchor the outer one so that as things swell, it tends to encourage lymph to go in. And as that is picked up, that lymph, there's less swelling and it closes the mini valve so lymph cannot leak out again. Are there questions on this? Okay, so once you got lymph into these lymphatic capillaries, how do you get it to move so that it'll eventually go into a lymph node and move towards the blood? What adaptation?
So typically lymph is moving slow and at a low pressure, and it actually doesn't have, the lymphatic system doesn't have a pump of its own, but it uses the force from the thoracic pump as well as the muscular pump to move the lymph through the nodes and back to closer to the larger veins. And then there's also valves in these vessels that prevent backflow and force the lymph in a one direction.
Very good. So that is, I mean, what you learned about veins is basically true for the larger lymphatic vessels. So they're being squeezed by thoracic and muscular pumps. Now, the muscular pumps we focused on for the blood circulation from veins were often skeletal muscles, right? And that's still true here. But if you're in a digestive tract, you could be talking about the smooth muscle of the digestive tract itself, because your digestive tract is constantly squeezing and that's pushing the lymph along as well. So you have muscular pumps, you have the thoracic pump, and as this lymph moves through the capillary, it eventually ends up in larger and larger vessels. And along the way, those vessels have lymph nodes so that the lymph has to enter into the lymph node. And I think the next question is about these lymph nodes, right? Who has that question? It's 9 on mine, but I think the numbers might be off with. Okay, Stephanie. So what happens with these lymphatic vessels at the lymph nodes, and where are the lymph nodes? Yes, where are the lymph nodes? Okay, sorry. Go ahead. Mm-hmm. Besides armpits, what other junctions do we have? Yes, so your neck and your oral cavity, So junctions are axillary. We're going back to 103A with these terms, right? So that's your armpits. Cervical, anywhere around your head. And your neck. And then how about where areas around your legs?
Gonadal.
Gonadal is, well, that's not really legs, but
The inguinal.
at least I hope not, unless it's really long. Yeah.
The inguinal area.
Inguinal area. There you go. So in lab today, there's going to be a map or today, or maybe, no, next week, maybe. There's a map we'll be seeing of where you find these junctions of lymph nodes. So MALT is any of those internal ones within the cavity itself, or as he said, gonadal areas, those are all part of your MALT system, your urogenital, your GI, your respiratory. But as far as lymph that's being collected around your arms and your legs, those all end up having, clusters of lymph nodes up at the junction. You don't really have lymph nodes in your arm itself or in your lower legs itself. You have most of them at the vast majority at the junctions. So, so one thing you might just relate to is if someone has breast cancer, they often remove lymph nodes in this area. They might remove lots of them. And what can happen is that will mean there's fewer avenues for lymph to eventually collect. And that person may have chronic edema in their arm because it's hard to get rid of the lymph from their arm. So, good. Okay. Before we go looking at the details of the lymph node and the thymus, let's talk about why you want to return this lymph. If you didn't return this lymph, that 2 millimeters, wherever I put it, that's coming out of mercury, right? I wrote on top of it, this 2 millimeters of mercury means you're chronically losing fluid out of your blood into your interstitial fluid and then picked up by the lymph. So the lymph is going to be being moved in order to ultimately bring that lymph back to the bloodstream. Before it gets there, you want to make sure it's gone through a lot of lymph nodes and be really filtered and any pathogens removed so that you're not sending the pathogens into your bloodstream. Now, where you do send it in, again, we'll do this next week, it'll be 2 structures called thoracic ducts. So that is a duct where the lymph is dumped into the blood, and they're found near the subclavian and jugular veins, fairly close to the heart. So ultimately, all that lymph that's collected is returned to the bloodstream so that you keep your blood volume and your interstitial volume in balance, that you're not having excess interstitial fluid. Okay? If you work standing up all the time, as I used to, then you might notice that at the end of the day, your feet are a little swollen. And that's because especially if you're standing, say, working a cash register or something, and you're not able to move around much. If you're not moving around, your muscular pump isn't as effective at getting the lymph up against gravity, and it can accumulate, and that can cause your feet to swell. So, so your chronic movement helps to return that lymph. Okay, the last 2, what time have we got? We've got time to finish at least these last couple questions in the lymphatic system. So when you do draw these next week, which I know is a ways off, but when you do draw them, the lymph node is kind of like this in a very crude way, right? It's got lymphatic vessels coming in, and those lymphatic vessels, maybe I'll draw them in a different color, have valves in them so that the flow will only go one way. The lymph flow will only go one way. into these lymph nodes. And then you'll notice that there's relatively few, maybe I'll draw those again in black, relatively few exits from this where the lymph comes out. There might only be 2 with, again, valves to prevent, so flow can only go one way through these. So like an earlier person mentioned, I can't remember who said it, it's going to pool in here. There are 2 layers, the cortex, oops, I did it again. This thing is very sensitive to my hand. Cortex, and the middle part's called the medulla. So with that in mind, who is doing the next to the last lymphatic about the function of the cortex versus the function of the medulla in a lymph node? Okay, Stephanie. Nice. Okay. So we did talk about antibodies when we did the blood typing. This is where they come from. B cells are mitotic in what's called a germinal center. So let me label that in my messy drawing. Germinal center. So you only see germinal centers when some B cell has been activated and undergoing mitosis. So you might see areas of the cortex that look fairly uniform, and then you'll see areas of the cortex where you see this dark rind with a light center. It's sort of donut shaped. And that's got a germinal center, which, as Stephanie mentioned, means that these B cells are generating plasma cells, and the plasma cells are releasing antibodies into the lymph. Eventually, that lymph makes it into the bloodstream, and from the bloodstream, antibodies are picked up and can be go out of the blood if there's inflammation into the interstitial fluid, and can battle things there, or they can battle infections in the blood itself. So antibodies, we're going to come back to a while from now when we do the adaptive response. But you're going to be drawing these germinal centers, so you need to know that there are B cells in the middle of those becoming plasma cells. Now, in order for the B cells to become a plasma cell, they need some T cells. So you do see T cells in the cortex as well. Okay, what about the medulla? What do you have in the medulla? And they have a name, antigen presenting cells. Again, more on this later, why they have to present the antigen. So we're going to call those guys APCs, antigen presenting cells. So when you look at the medulla in the slides next week, you'll see that it's a little less dense in its staining. So there's a lot more places that the lymph is pooling in here. This entire lymph node is made of reticular connective tissue, except for the very outer part, which we'll call the capsule. And that capsule is just keeping it separate from the surrounding tissue. So these this medulla, the whole thing is reticular, but this is dense in cells, lots and lots and lots of B and T cells. This has fewer cells, but the cells that are there are dendritic and macrophages, and a lot more room for interstitial fluid to kind of pool in here for these guys to phagocytize the potential pathogen and then present it to the B and T cells as Stephanie described. All right, so that's lymph nodes. Now, one of the things you'll see next week in lab is that lymphoid tissue is basically like the cortex of a lymph node functionally. It's going to have B cells, T cells, and you're going to potentially have germinal centers in there. Even the white pulp in the spleen is basically like having some of the cortex functionally of a lymph node, but now embedded in the spleen. You'll have B cells, they can make germinal centers, and T cells. So then what about the medulla? Well, associated with MALT, you have, and with the red pulp in the spleen, you have dendritic cells and macrophages that fulfill the role of the medulla. They're just dispersed through the tissue more rather than being sort of in a central location. So these terms, cortex and medulla, obviously you're familiar with from the brain. Luckily, this kind of fits what you know, the cortex on the outside, the medulla in the middle. Well, the spleen has a cortex and a medulla, but they're not functionally the same as the cortex and the medulla of the lymph node. They're just physically, the cortex is more on the outside, the medulla is in the middle. Same with the thymus. They're going to be cortexes and medullas, but again, they're not physically the same. They're not going to have, the thymus isn't going to have any B cells, so anywhere. So they're going to kind of loosely use these terms for just the arrangement rather than what functions they're serving. Okay, how are we doing on time here? We have 5 minutes. Let's finish these up, and as I thought might happen, we won't get to the first immune cells system questions. So what about the thymus? Is that you still, Stephanie?
It's going to be me.
Okay.
Kaylin.
Who is this? Kaylin?
Kaylin.
Okay, what about the thymus?
The T cells mature in the thymus and go through a selection process. And in part of this selection process, it involves eliminating some T cells through apoptosis. And for apoptosis, the cell contains lysosomes and digestive enzymes that are released so that the cell can kill itself.
Okay. And one of the ways in which this occurs is the thymus has to generate some hormones, right? For this process to occur. So the thymus, unlike the other lymphatic organs, contains not just reticular connective tissue, but contains epithelial cells. You won't have to identify them, but they make hormones, and there are 2 of them. Don't worry about their names because there's just one less name to learn. I don't even think they name both of them in the book. But they help the maturation. They're required for the maturation of the T cells, as she brought out. And they're useful for this selection. So we will come back to what happens in the thymus, but you're going to have positive and negative selection. While you're doing this is you're trying to get rid of T cells that either recognize antigens on your own body cells so you don't attack your own body cells. And you also want to make sure that the T cells you generate bind their antigen strongly. So if you have some, namby-pamby T cell that doesn't bind its antigen very well, get rid of it and let the bone marrow make more and, make a better selection of strongly binding T cells that then also don't recognize your own antigens. So this is all honing your immune system, making it work well. There are people, however, that often they think associated, but some we don't know, with a previous viral infection. There's some types of autoimmune responses where you do generate T cells, or in some cases you generate antibodies, so inappropriate antibodies from B cells that attack your own body system. So very quickly, we're going to start the immune system next time, and we're going to start talking about how the immune system can recognize self from non-self. And the adaptive is extremely specific in how it decides to activate a B or a T cell. And so you definitely would like to get rid of any recognition of self, since some of these T cells in particular are going to kill body cells. That's going to be what the fate of some of the T cells are, to seek out and kill body cells that have been infected by a virus or a cancer. And so you definitely want to hone the T cells very well so that you're sure that they're not inappropriately killing your own body cells that are normal, perfectly healthy cells, which can happen in some autoimmune conditions where people do have that happening and they're killing their own body cells. All right, so when we come back, we're going to have that group start talking about the innate, what we'll call the innate versus the adaptive immune response in a big overview of the immune system. So most of the rest of this lymphatic stuff we're going to be doing in lab and be tested in the lab. As you can see, I already referred to some of the slides that you're going to be drawing. But some of it is hard to test in the lab, like the mini valves and things like that. So it could show up in a future lecture Canvas quiz. So one heads up on the immune system. I do follow the book fairly closely, but I go into, in a couple places, I go into a little more detail, kind of like we did in the cardiac action potential. The biggest challenge of the immune system is its challenge and its strength is it's extremely complex. It's very easy to miss the forest for the trees. So we're going to start by a big overview, which the book does too. And there are many things that I've put into useful for lecture, or not the useful for lecture, I think it's called lecture exam 2 folder. Their folder in there to help you keep a lot of these things straight, because it's very easy to get lost in the details of it. So I would spend a good amount of time really listening to those lectures and then looking at the handouts that are in Canvas to help you so you don't get too lost. The other challenge is it's molecular. So we've been dealing with, you can kind of visualize a heart beating or the volume changing in the chest wall. You can visualize a lot of the stuff that's going on in a gross anatomy sort of way. Whereas here, we're really talking about cellular level and molecular level and how those things are interacting. So it's a little harder to mentally picture. So again, just a warning on that. All right, I am going to let you go if there are no questions. And in the lab today, remember you're drawing some white blood cells, and then you're going to be in groups, and I posted those groups for an assignment on a differential white blood cell analysis. And that stuff isn't going to be due until after the lab test. So this gives you, some time, since this is obviously on the second lab test, this material we're doing in lab today, to turn it in. So make sure you pay attention. I believe on the syllabus, I have this due on the 8th. So you're going to be turning in the handout as well as the drawings. I neglected to write that in the list, but you'll be turning in your handout. There's only like one question on it. It's not very complex. Okay, so I'll let you go and meet you in lab in about 15 minutes. Are there any questions? Let me see if there's anything in chat. Okay, let's see. What happens in lupus? Somebody asked, and well, let's not worry about that one. But what happens in lupus? Well, that's one of the ones we really don't know. It is an autoimmune condition, but we really don't know what is triggering that, if that's what you're asking by what happens in lupus. And people get inappropriate immune responses, which cause inflammation, is my understanding of lupus, but I'd have to really look it up. It's not a fun thing to happen. It does seem to happen more in women than men. And so there may be hormonal, exaggerations of this condition, but I'm not sure they really know why that happens. Then we have another question from Manreed. Are we supposed to watch lecture 10 and 11? Are those the immune system ones? Because I don't have the numbers. I just remember the names. I don't have them in front of me. What I would watch is make sure you've watched the lymphatic one for today, obviously, and then the innate one, at least the first innate one by Tuesday, because I know you're studying for the lab test. So watch that first innate one by Tuesday. And then if you haven't watched the second innate one, that's probably fine, because I'm not going to rush through the immune system. It's too complicated. Okay, I'm sad to hear that your mother has lupus. That's not an easy condition. Okay, so why don't I see you in about 15 minutes, and